Good morning, guys. Welcome to another Biology with Miss Cherry. As you can see today, we're moving away just temporarily from the isolation evolution kind of stuff. So have a go at this question for the beginning. What is the graph showing? Why does the y-axis value change? You might notice already before even spending too much time looking at the graph, we're losing the log number. And if you're not sure what the log number is, we're going to go through it. And then finally, what brings around that change? So why do we have such a significant change in number of cells? What do you think this curve could be of? And why do we get those different phases? So just have a think about this. We're going to go through this later on in the lesson, so we won't spend some time going over this as part of the starter now. So pause, start your thoughts about the lesson now for me, and then play again in a few minutes once you've done these questions. So today we are looking at variation in population size, and we're going to be looking at biotic and abiotic factors. We're going to be looking at growth curves because there's a very high chance that they might bring in some kind of graph kind of questions with these topics, and just see how and why populations change over time. This is the start of a few lessons looking at the factors influencing population sizes. This is more the numerical basis and more of a general overview rather than specific. So over the next few lessons, we'll look at things like competition, predation, things like that. Today, we're just looking at a big, broad overview. So let's get started. In your notes for me, have a go at defining the word populations and what are the two different types of factor that can affect population size. Now you can think about this, you can make some notes and you can pause and shut me up for a second, or we can just get straight into it. And a population is a group of individuals of the same species. Key. It's not anything like an ecosystem. We're not talking about the interaction between organisms now. We're looking at individual groups of humans, cats, elephants, that are all in the same place at the same time. And that same time is key because we know that populations can grow and shrink at a moment's notice. And these factors affecting populations can be either biotic or abiotic. And we had a look at these when we went through the title and the learning objectives. Biotic factors are those which are living, whereas abiotic are non-living factors. And can you think of an example of each? We've had a talk about a few of the biotic factors so far today, but abiotic is potentially even more important for the survival of a species. Now we've said that population sizes can vary. Okay, we know that when we're looking at populations, we're looking at a group of organisms of the same species that occupy the same environment at a particular time, and that's because population sizes change, so we have to be specific. And the whole idea about population size is related to how the births compare to the deaths. So if we've got a higher death rate than birth, then we get a lower population size because more individuals are dying. Whereas if we get more births than deaths, then we get a bigger population size. And for this reason, we say that populations are dynamic. When we're talking about a population, we're looking at a very specific snapshot in time. At this moment, say at, what is it today? It's the 8th of February. So at 7:14 on the 8th of February, Miss Cherry's house is populated by one human. That's it. No one's died, I hope. But, you get my gist. At this moment in time, if you were to ask me in about 12 hours, there'd probably be 2 people here. It's all fluctuating. There's variable numbers. Now, to show populations, we generally use a scatter graph. Okay, so individuals which grow quite slowly, most mammals, where the population size isn't going to vary too much over time, we just show them on a standard scatter graph. Okay, so we've just got a standard scale up the size. Normally you'd put a unit or something, maybe thousands if you don't want to squeeze in a bajillion zeros on that axis. But please, 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 do not do a dot to dot. Do not do a dot to dot with a scatter graph because that will be a mess. We're not looking for a jaggedy line that shows the pattern year on year. No, we are looking for the line of best fit, the line that best fits the data that we've got, not multiple lines joined together in a dot to dot. So now it's your turn to do some plotting. I've got some data here, so the years on the X and the number of lions in the wild to show the growth rate of lions in the wild population since 1980. You can either do this on Excel, you can do it on lined paper and take photos and put it into your notes, or you can Google graph paper and print out a couple of sheets of graph paper, because you're going to need some in a sec as well, and plot this graph for me. And have a think, when you're done, it should only take you a few minutes, what factors might cause the temporary decline. So there's some growth and decline rates, or growth and decline periods on this graph. What could have caused them? Maybe there's a historical basis. I don't want you to look too deeply into it, but knowing the background and considering the background of a population is key sometimes. Some organisms, on the other hand, don't grow slowly. Some grow really, really, really quickly. Things like bacteria, and instead of using a numerical scale such as like 5, 10, 15, 20, 25,000 bacteria, we've got to use a different scale. Okay, those going up in standard increments is not going to be accurate enough to relay the population size at that time. So instead we have to use something called the log scale, and we're going to look quickly at what that means. So the log scale is a way of showing exponential data. So we don't need to know how to change normal numbers into log numbers. You don't need to worry about that. If they do, however, give you a data table that has log data in it, all you've got to do is plot that data as if it was a normal number, and make sure that you put number of bacteria and then you put log next to it to highlight the fact that you know that it's a different value to what it should actually realistically be. So the log value is able to make a really big number quite small. It's to do with condensing it down to the base of 10 or something like that, I think. If you do A-level maths, this will probably be quite standard for you. If you don't, just treat it like a normal number. So have a go at this. As you can see, we have got time in hours, number of bacteria, and the log number of bacteria. And even looking at this graph, you can see that 50 is equal to log 1.7, whereas 31,500 is equal to log 4.5. So you can see now already that the values of bacteria is much easier to plot as the log values, just because they're smaller, they look more regular. So have a go at plotting the data and answer those two questions at the bottom. When is bacterial growth the fastest and why? And what happens to bacterial growth after 8 hours and why? This is the last part of data plotting for the rest of the lesson. Please spend your time making it look neat. If you're not sure what it should look like, ask me. We can discuss the answers to questions 1 and 2. Just pop up on the chat and I'm here if you need me. So the limiting factors are things that we're going to spend a little bit more time looking at now over the next few slides or so. And the limiting factor is a constraint of the environment that is going to limit that population growth. Now, generally in a system that is open, i.e. things can move between places, we see a period in populations of really rapid growth. We almost call it exponential growth, although it does stop and slow. And after that time, that initial exponential growth, so there'd be a rapid upshot, a positive gradient in a graph, then we get a time where either the growth rate slows or even stops completely, because external factors are going to inhibit population growth. And this is where we've got something called the carrying capacity inactivity. And this is the maximum population size an environment can support with resources, so positively supporting, due to the presence of other limiting factors. So something else in the environment that's stopping the further growth of my population. And these two key terms, limiting factor and carrying capacity, are really, really important to be able to use. So if you're not too sure, maybe take a look at the online revision guide that I posted, maybe take a look at some online resources and see how others use the terms limiting factor and carrying capacity within a sentence. So let's have a think about this example. You are a single photosynthetic bacterium bunged in a tank, and it's summertime. Okay, you're outside maybe, there's lots of light, lots of nutrients, and the temperature of the bath that you're in, the little water bath, is about 15 degrees C. How quickly will you initially multiply to increase your bacterial population? And by that I mean, will it be fast, will it be slow? And what will happen to limit the growth of your population over time? So I want you to pause now and think about maybe using those key terms that we just had a look at to this question. Let's try and apply them. So pause me and play again when you're ready to look at the answers. Now initially, you should have said that there will be a phase of exponential growth because there are no limiting factors. However, when the population reaches the carrying capacity, then we've got the influence of those limiting factors in action. Okay, so then growth is going to slow or stop. Maybe it will even decline. But the decline is only going to happen when deaths are more than births. Or divisions, okay? Remember, we're talking about bacteria here. So as we've said, over time, rapid exponential growth, got that asexual reproduction, binary fission. Have a look back at module 2 if you're not too sure what this means. And over time, the growth rate slows and the population increase isn't as rapid, if at all. Now I've put 4 factors at the bottom: mineral iron levels, production of a lawn of bacteria on the tank surface, introduction of other species, and winter. And I would like you to have a go at explaining how each of those limits bacterial growth. So you're going to pause. I imagine it will probably take about 5 minutes. So how each of those limits the population. It's an explain question. Use your knowledge. Remember, you are a photosynthetic bacterium. Think about what that would mean for things like your coloration, where you are, what kind of temperatures you might need, and why. So again, pause, spend about 5 minutes on this, and play again when you're ready to go through the answers. So let's take a look at this first factor then. Mineral iron levels initially facilitate a rapid increase in bacterial growth. They're going to take up the minerals for reproduction. However, over time, as those bacteria keep reproducing, the mineral ions deplete. They're being used up. And the use of these finite resources, okay, so they can run out, means that over time, there's going to be less mineral ions available for immature bacteria. And I want to draw attention to the fact that we're saying immature bacteria. They're not baby bacteria, because bacteria are bacteria. Why would we make them babies? You know, they're all the same. They're just bigger or smaller. They haven't quite developed as much as the mature bacteria, but they're still just a bacterium. Now for the second one, how does that lawn of bacteria on the tank surface affect growth, inhibit growth? And this is all to do with the fact that you are a photosynthetic bacteria. So if you can't get any sunlight, because there's a lawn of bacteria, or a sheet almost, so a sheet of bacteria effectively grows over the surface of the tank, that's going to block the light. And if it blocks the light, then bacteria that are potentially further deeper down in the tank aren't going to be able to have that light to undergo photosynthesis. They're not going to get glucose. They're not going to be able to get energy through respiration. They're not going to be able to reproduce. You're limiting your numbers. If we introduce another species, maybe a predator, maybe just another different type of bacterium, they could provide some form of competition. Okay, so now we're starting to look at how biotic factors are going to influence. So competition, when they're trying to use the same resources as our pre-existing bacterium. Alternatively, they could also be a predator. So maybe they're actually going to feed off of that photosynthetic bacteria and further limit the numbers. Now winter is really significant. This is a problem for lots of organisms when the temperatures get colder, especially if you're unicellular. So the lower the temperature, the less kinetic energy we've got. Okay, standard answer. We know that temperature is going to affect the energy of our molecules, and therefore, we're thinking back to our basic GCSE level enzymes kind of stuff. The less energy in our molecules, the less likely we are to get successful collisions between the enzymes and the substrates in that photosynthetic reaction. Remember, we've got loads of enzymes. It's an enzyme-controlled Calvin cycle. If we don't have that in the Calvin cycle, we're not going to have anything, are we? Because it's not going to keep producing RUBP and all of that good stuff to keep that Calvin cycle going on and on and on again. As well as this, in winter there's less light levels, isn't there? Even if you look outside now, it's quite dim, quite gloomy. That lower light levels means that you're going to have a less light-dependent reaction. Okay, photosynthesis is not going to occur as quickly. And so overall, we get less glucose produced, which means that we've got less energy available for cell division via respiration. You see now how we're starting to link many topics. So we've got module 1 here. We've got GCSE when we're talking about collisions. We've got module 6 or module 5. We've got cell division, so we're back to module 2 when we're looking at binary fission. We've got respiration, module 5 again. We're linking everything together now. This is the final few steps for us to see that actually everything that we've learned individually builds on top of each other. Now I'd like you to spend some time having a look, doing some research at how each of these abiotic factors, so temperature, light, pH, water availability, oxygen availability, and edaphic factors, which you might not know what it means, but I'd like you to have a look and a research, explain how each of them might affect a population of organisms. I'd like you to be as detailed as possible, so you're going to need to do some research. And if you want to, for each factor, explain how an organism might become adapted to reduce the impact or the effect of those abiotic factors. So there's 1, 2, 3, 4, 5, 6. You should probably take about 10 to 15 minutes doing this. So pause and play again when you're ready to go through them. Now the first factor, light, has a significant impact on plant growth. And we've already seen that winter has a big effect on the availability of light and therefore the growth of plants. But the greater the availability of light, so maybe they're in a nice wide open field, the greater success a plant species is going to have. And this is purely because of photosynthesis. So we're always looking to see how that light energy is going to be able to be transformed into photolysis, but also how that light energy is going to be able to provide energy for molecules to move, but also the importance of any other energy that come with that light energy. So remember that things like the sun provide radiation, which is going to be a kind of thermal influence on the molecules as well. Now adaptations for low light conditions, maybe plants is going to have some large leaves. Maybe they're going to be quite big and broad, maybe like sunflowers, to maximise the surface area available for uptake. Maybe they're going to have differently orientated palisade mesophyll cells, so all of the cells that are specialised for light uptake because of those chloroplasts. Perhaps they might have different photosynthetic pigments which require less light. So we know that in our standard examples of photosynthesis, we saw that we've got a 680 nanometer pigment that absorbs that wavelength of light, and also a 700 nanometer pigment which absorbs that wavelength of light. But perhaps we have low light conditions, so we can't use those pigments. And over time, plants might have developed different colorations in their leaves, and therefore different colorations in their pigments to absorb a different wavelength of light that's maybe more readily accessible. And finally, they might have adapted to have reproductive systems to operate at this low light intensity, but at this low light intensity, those reproductive systems are now optimal. They work the most efficiently. Temperature, very similar to light, so it's going to control our enzymatic reactions, our enzyme controlled reactions, metabolism and things like that. We're going to have an impact on plants again. So obviously the warmer it is, the more plants are going to develop, they're going to grow. You can see that in the growth in your garden from spring, or transitioning from winter to spring to summer. But also in ectothermic animals, so animals that rely on the outside environment to control their body temperatures. In other organisms, the changes in temperatures are going to trigger movement changes, so actual physiological changes, behaviour changes, such as migration patterns. So maybe the chilling of the temperature means that these birds are going to fly now from England to South America if they can get across the ocean. Maybe in certain circumstances, if you're a bear, for example, you might start hibernating, or plants might start losing their leaves. Important to remember as well that the temperature and the light intensity sort of go hand in hand at this point. So the lower the light intensity, generally the lower the temperature, because we're in winter, and therefore there's going to be no need to have the leaves anymore in some plants because they're not going to be able to photosynthesize. So instead of trying to hold on to them, the plant just goes, nah, get rid, and chucks them off. Adaptations though, this is where we're going to look at ectotherms. So if we were looking at the OCR specification, we would have done a whole lesson on ectotherms, or organisms that depend on their external environment to regulate their temperature. Things like desert organisms, lizards, lots of reptiles change their behaviours depending on what their environment is and how they need to respond to it. So things like a lizard are going to bask in the sun or bask in the sun if it's cold, and that way they can warm up their body, so increase body temp, and they're going to be able to increase the activity of their enzymes. So they're going to be more metabolically active in the hot. Whereas if they get too hot and they need to start cooling down, then they will go under a rock, and they're just going to reduce their body temperature slowly, passively as well. They're not doing anything to do it. They're just getting under a rock and they're just going to chill to start cooling down. pH, another significant factor impacting organisms, mainly because of its impact on enzyme activity. Now, where populations are bigger in the world, it's because the environment has an optimum pH. So maybe if they're aquatic species, there's going to be maybe an acidic or alkaline water that suits the enzymes of the organisms that live there. However, there are some organisms called extremophiles. Xerophytes are examples of this. If you've done Taq polymerase in the PCR lesson for Module 8, that's an example of an extremophile. And extremophiles are organisms that have occupied niches, so small areas of the environment where nothing else really lives, and they've adapted to them. Things like geothermal vents, where your Taq or Thermus aquaticus bacteria lives. This is the one that you use in the polymerase chain reaction. These geothermal vents have got really, really low pHs. They're very challenging situations for any other organisms to live in. And in doing that, they avoid competition. So they're effectively hiding without hiding. Water availability is going to affect all organisms quite significantly. Okay? A lack of water is going to mean that we've got a stress on our system. So if plants don't have water, obviously they're going to become quite flaccid, and maybe they're not going to be able to hold themselves up to photosynthesize. If animals don't have enough water, then obviously they're just going to dehydrate, and that's going to not be a good time. However, remember back to Module 3, xerophytes, things like cacti, things like marram grass, all of those organisms that live in waterlocked situations, so the Arctic, desert environments, the beach where it's highly salty, are going to develop strategies to cope with that water stress. And I want you to think back to Module 3. Can you tell me any adaptations of xerophytes that mean that they're able to survive low water conditions? So you might want to pause and look back again. There's a YouTube video called Water Lot in Plants. It's Module 3. It is up there if you wanted to go back and have a look at them. Oxygen availability is going to impact aquatic ecosystems, so underwater ecosystems like fish and all of the plants that live under the sea as well. A fun fact, fast flowing cold water carries more oxygen than warm, slow-flowing water. And that warm, slow-flowing water, so maybe tropical streams, for example, is going to reduce the amount of oxygen that we've got available to the organisms. And over time, that could lead to suffocation. And we can see the impact and the importance of oxygen dissolved in water when we look back to Module 3 and we look back to the countercurrent flow in fish. And again, if you're not too sure about that, there's a video for it. It's called Gas Exchange in Fish. I think it's like Module 3, Lesson 3, I think. But also, we've got to think about how oxygen availability impacts plants. So waterlogged soil that's got lots of water, if you've overwatered the plants, or maybe we're in a kind of boggy situation like rice fields, paddy fields, things like that, is going to reduce the oxygen availability for plants. The spaces between soil particles are filled with water, but it's stagnant. So the water doesn't move, and that means that it just sits there permeating into the soil structure. However, certain plants, things like rice plants in paddy fields, have been adapted to germinate under these low oxygen conditions. And you might want to read this article, it's quite interesting. Rice plants don't respond to low oxygen situations in the same way that other plants do. In fact, these rice plants don't actually see low oxygen conditions as a stress anymore. What they do is they use it and they thrive. And that again is an example of an organism adapting to a niche situation. Now finally, the edaphic or soil-based factors. Soils are very, very different. I don't know if you tried digging in your garden, maybe versus my garden, maybe a garden down on the South Dorset coast. There's going to be very different soil compositions. And the size of the particles and the makeup of the particles is very, very different in different areas. And this impacts what we can grow in them. So if we've got quite a clayey, sticky soil, then we've got quite fine particles that clump together when they're wet. And you'll know if you've got clayey soil, it might be slightly redder. And this means that it's easily waterlogged. The clay holds on to the water very, very well. It provides quite a squishy, clumpy texture that you can form into big balls. Loam is when we've got different sized particles and it can hold on to water, but it doesn't waterlog. So the water can sit there and provide nutrients and transport oxygen, but it doesn't just remain stagnant, a bit like in those paddy fields that we looked at on the previous slide. And finally, sandy soil is quite coarse, so the particles are quite big, but they're well separated, and that means that water can flow freely through it, but doesn't necessarily mean that it's going to be able to hold on to enough water to provide plants with nutrients and oxygen. Now it's your turn. Have a go at the tasks on page 208 to 209. There's photos on OneNote, including the growth and size of human populations. You're going to need to show all your workings out nice and clearly, and then you are going to self-assess, because we're at home and you can't touch anyone, using the photos that I've put on OneNote again with the answers. Now also on OneNote there is a page of exam questions. Could you please have a go at answering them as best as you can? They're all about populations and things like that. And I'm here if you've got any questions. Now answers wise, I'm just going to be quiet because I don't have the questions in front of me. So you can pause now and you can have a look at your marks, see how you've gone, see where you went wrong, and let me know if you've got any questions. And the final set of marks. So you should have a total of out of 14. So give yourself a score, see how you did, and I hope you found that useful. And I'll see you next time, starting to look a little bit more detail at populations and the factors affecting them biotically. Bye bye.